discuss the more detail, more towards the anatomical point of view of how the biosignal are generated within our body. Okay. Wait. So the objective of this chapter is that you will be able to understand how the biosignal is generated in the body at the different level of physiological structure. Yeah, many are actually generated even at the cell level. Yeah, the electrocardiograph, electromyograph, electroencephalograph. These are all the bioelectric signal. They are actually generated within the cell structure and then spreading among the cells to be generated as in the single bioelectric signal. Yeah, yeah. Some are generated in the tissue level, some are organ. Then to know the physical and biochemical mechanism related to the generation of this biosignal at the different physiological structure. So we will reviewing some of the material that you may have learned from cytology and histology, I believe. Yeah. Although some you will be learning in the anatomy and physiology in the next semester, but mostly the emphasis will be in cell and neuron and receptor, and then maybe muscle and heart. Okay, so then the next chapter we will be already dealing with the sensors to detect this physiological phenomena using the sensors, yeah, to detect the biosignal. So cells, what are the functional structures within cell? How can they generate a biosignal? Then actually the most important part in the cells that is related with biosignal generation. So far, what we have known, you know, we here the scientists, are the cell membrane and the cell membrane potential. It is the cell membrane potential that is responsible to generate what we eventually, or the physician eventually measure from outside the body as the bioelectricity, yeah, ECG, EEG, EOG, EMG, etc. Then how does the cell membrane potential propagate? Yeah, propagate of the excitation. So these are the structures of cell. Um, what is number one?
Nucleus.
Remember? Still remember from last semester?
Nucleolus inti.
Nucleolus.
Nucleolus.
Number two?
Nucleus.
Nucleus. Number three, here. Or the red spot, the red dot.
Ribosome. Hello. Check.
Number nine. What is number nine?
Mitochondria.
What, Anita?
Mitochondria.
Mitochondria. Yeah. Okay. Let's check the answer. This is the answer. So number one, yes, correct. Nucleolus. Number two is the nucleus. The nucleus is the dense structure inside the nucleus that produces the ribonucleic acids, yeah, the RNA needed for the protein synthesis in the ribosome. Number two is the nucleus. No, this is the one that contains our genetic information. Number three is the synthesis of the protein, the ribosome, yeah, the red dot here, which is located in number five. Number five is the rough endoplasmic reticulum. Number four, where is number four? Is reticulum. Here is the vesicle, yeah, the vesicle body. Usually, it is used to secrete waste from the metabolism. Number six is the Golgi apparatus. Where is number six? This part, yeah, without any red dots, so similar like the rough in the endoplasmic reticulum. The Golgi apparatus absorbs, modifies, and packages macromolecules into vesicles. Yeah, vesicles is the small fatty acids. Yeah. Enclosement. Membrane enclosed sac and is involved in cell secretion. Number 7 is cytoskeleton. Where is number 7? So this one, well, actually, it's not located like this one. Cytoskeleton is located all around the cells to give the structure of the cells. Yeah. If you listen to the presentation by Professor Kinoka last Friday, yeah, the cytoskeleton can form also legs. Yeah, yeah, this is something new also that can make the cells to be moving. Yeah, in the side their cell culture moving around, and then also making connection with other cells. Then smooth endoplasmic reticulum, where many different metabolic reactions occur. That is, for example, the production of hormones in number 8. Where is number 8? This one. Yeah. This is smooth, so it means there is no ribosomes. It is connected with the rough endoplasmic reticulum, but there is no ribosome attached to the smooth endoplasmic reticulum. Then mitochondria, as what Anita mentioned, and the plural is mitochondria. That is the cellular power plants generating the ATP, adenosine triphosphate, the energy substance, common source of chemical energy within the cell. Number 10 is vacuole. This is vacuole, kind of a cavity, a small fluid containing vesicle, holding the inorganic and organic molecules, mainly assist in extrusion of macromolecules from the cell and in the engulfment of foreign substances. Then cytosol. Cytosol is basically the liquid within the body or cytoplasmic liquid. Yeah. Then lysosome, number 12 here, smaller than the vacuole. Then lysosome, number 12, the digestive organ of the cell, which contain enzyme to decompose outdated organelles and engulf foreign substances. Then 13 is the centriole. Yeah, the centriole. Okay, so this is how under microscopy how the cells look like. This is images of different mammalian cells. The mammalian cells means like actually our human, we are mammalian as well. So our cells basically look like this. So A is the monocyte in mouse spleen, yeah, in mouse. Excuse me. So the B is epithelial cell from the mouse kidney. So here you can see the different organelles located within this mammalian cell. Yeah, in the mouse spleen, the nucleus looks very large here, and we see here the endoplasmic reticulum, where it has ribosomes. Then the Golgi apparatus, the mitochondria here, and this is the space between the different cells, the extracellular space. Okay, and here in the kidney, it has these additional structures called as the microphyli, which is whose shapes is like hairs, yes, tiny hairs protruding from the cell's body. And this is one of the most important part, especially in biosignal generation, the cell membrane. Yeah? Basically, a cell membrane consists of bilayer membrane structure because bilayer membrane, one layer consists of a phospholipid structure. Yeah? This is called, that's why it is called the phospholipid bilayer. So one single structure here. Yeah, let me go here. This one is the phospholipid, one single phospholipid bilayer. This one is another phospholipid bilayer, and this part is the fatty acid part. Yeah, the tails. They are the fatty acid, which is very hydrophobic. That's why they are connected to each other. While this part is the phosphorus part, which is hydrophilic, and that's why they are facing the extracellular fluid, and the other part facing towards the intracellular fluid or the cytoplasma. Yeah. So within the membrane itself, within the bilayer lipids or phospholipid bilayer membrane itself, it contains a lot of things. Yeah, it contains protein. Here, the integral protein, it contains also glycoprotein, a combination of protein and glyco means from sugar, yeah, polysaccharide, yeah, and it contains the globular protein. It contains also this one, protein channel, which is responsible in transporting ions from and to the cells. Yeah, that's why, and the ions exchange between the intracellular part of the cell and the extracellular part is responsible for the electric generation, yeah, the ionic signal generation. And here we see, what is this? Long alpha helix, yeah, alpha helix protein. This is also an integral protein. There is also many glycolipid here. Yeah, this is glycolipid, in which the lower part here contains lipid, which is then integrated with the other lipid parts of the bilayer membrane, and the outer part contains the glyco chain, yeah, the polysaccharide chain. And here are the cytoskeleton, the filament of the cytoskeleton that form the structure of the cell. So the bilayer membrane spontaneously results from electrostatic interaction, yeah, between the aqueous milieu, full of the ional and polar structure, which is this part, yeah, the phosphorus part. Then the electrostatic behavior of the lipid molecule, the inner part, include both polar and non-polar structure. So this is how the molecular structure of bilayer phospholipid bilayer. So the round part in the previous slide is basically this choline phosphate glycerol structure. The choline structure contains this amino acid functional group. Then it has this phosphate structure in the middle, which is very polar, that it can make hydrogen bonding with the surrounding water, either in the extracellular fluid or in the cytoplasma. And we have the glycerol part, which is also polar because it contains a lot of oxygen. So oxygen usually contains negative dipole, if you remember your chemistry. Then the tails, the two tails, are basically fatty acids. Yeah, fatty acid is basically contain only C and H, long fatty acids. Yeah, this remember the CH structure. So here you know that it is not polar. Yeah, there is no dipole inside. Yeah, so because and that is why it is non-polar region of this phospholipid structure. They will bind to each other inside the phospholipid bilayer, yeah, in the middle part. Okay. Then this is a simplified illustration. So you see here the polar structure, the hydrophilic structure will mix hydrogen bonding, electrostatic interaction, or van der Waals interaction, you may say like that, with the surrounding water molecule in the extracellular fluid or in the intracellular cytoplasma. See here, because the positive part, positive charge here, yeah, from the choline functional group will bind to the negative charge of the water molecule, which is located in the oxygen. Yeah, and the phosphate functional group here, which is very negative, will bind to the positive charge, which is the hydrogen part of the water molecule. So here contain. So this is water molecule. There is E, this is the electric field, and D, the electric flux density. So basically, an electrostatic attraction will occur between the ional and polar structures of inverse sign, whereas the resulting force magnitude is proportional to the energy W of electric stray field in between the attracting structure with the electric field magnitude E and electric flux density magnitude D. So the energy, the minimal energy will be given in this structure, the integral over volume, E times D divided by 2 dV around that volume. Or we can change the E as D divided by epsilon. Epsilon is the, remember the capacitance? Yeah, so this is the electric permittivity of the medium. So the membrane's protein, there are as I have explained in the previous slide, many proteins attach on the membrane, inside the membrane itself, that there are receptors for hormones, also enzymes, antigens, yeah, which will react or combine with the corresponding antibody. Glycoprotein, and the glycoprotein is which is linked with carbohydrates, protein that is linked with carbohydrates, and they are responsible for giving negative charges to the outer surface of the cell. So the bonding, and as you maybe have learned also in biochemistry, and I think this semester you are learning from enzyme biochemistry as well. The bonding between two or multiple molecules is likely to occur only if these two conditions are fulfilled. Complementary charges or complementary polar position exist on both interacting sides of the molecule. Means one is positive, the other one is negatively charged. So the attracting electrostatic forces between the molecules favor bonding. Then after bonding, the energy of these electric stray fields is reduced because of mutual saturation of the fields. Yeah, because there will be they bind, but then there will be repulsive forces also to compensate to saturate the binding. Furthermore, the close proximity of the nonpolar regions of both molecules favors their bonding because the mutual saturation of the charged regions elsewhere is not disturbed. So not only complementary charges are important in the binding between biomolecules, yeah, you can ask Pak Hollis later about this, about biochemistry, but also the complementary shapes of the interacting molecular site. That they will be promoting the bonding as well to the best geometrical lock match. Yeah, the lock and key concept in enzyme interaction, also in antibody antigen interaction. Yeah, the lock and key concept is one of the key concept in biochemistry. Yeah, so molecular tips fit into molecular notches. So remember this. There are two important mechanisms: the key lock and key concept, or complementary shapes, and the complementary charges or complementary polar position. This is something which happened in the phospholipid biolayer. So this is the lock and key concept. So they need to be, so for example, this part needs to bind this part, and this part also can bind to this notch part, etc. Yeah, so when they close, they complement to each other, become a complete binding. But then it has to be supported also by the complementary charges. So this part is negatively charged, this part is positively charged. So there will be an attractive electrostatic force between these two parts. Yeah. So the binding occurs between the molecules. Bonding here is electrostatic, or dipole or van der Waals, something like that, not covalent or ionic, because they are not molecular binding, but instead supramolecular binding. So temporary binding. So there is also, they can be dissociated using the correct condition, for example, using heat or other things. So, so the enzyme itself here, usually in the biochemical system inside our body in cells, will function as catalysts, yeah, to provide the correct binding site. So that's why this part will bind first to this, and this part will bind first here, and this will promote the binding between the two. After the two binding, usually with enzyme catalysis, then this will dissociate. This product, this new product, will dissociate from the enzyme. Yeah, for example, like glucose oxidase, oxidize glucose into glucagon, for example. The charge and shape complementary in terms of lock and key concept, which assists the chemical bonding of molecule. In addition, an enzyme molecule may accelerate the bonding process. This is before bonding and after binding. Okay, so apparently I have been reminded that the time is up. Okay, so that will be all for today.